Welcome back to Anatomy and Physiology on Catalyst University. My name is Kevin Tokoph. Please make sure to like this video and subscribe to my channel for future videos and notifications. In this video, we're going to talk about graded potentials in the context of neurons. All right, so with graded potentials, if you look at this in a textbook or your anatomy class, you'll often see this table that'll compare graded potentials to action potentials. For example, graded potentials vary in size, action potentials are an all or none phenomenon, and so on and so forth. And that's good to know. But really in this video, we're going to focus on the big picture of graded potentials. What are they? Because I think sometimes we get lost in like little bitty facts about graded potentials, comparing them to action potentials, and you lose out on really what they are. Okay, so we're going to talk about that here. And before we do that, we need to talk briefly about what's called resting membrane potential. Okay, and also what we have here in terms of the neuron. So this is the cell body of a neuron right here, okay, represented by this circle. This is the cell body, also called the soma. In fact, you'll also see graded potentials occur in both the dendrites and the cell body. Okay, this arrow over here represents the axon. Okay, graded potentials do not occur in the axon, so we're restricting ourselves to the dendrites and cell body, but I've just shown the cell body here for simplicity. Okay. Also, there are these proteins in the membranes, and I'll explain what these are specifically in a little bit, but it suffices to say for now that these proteins represent both receptors and ion channels. Okay, so make sure you bear that in mind. Now, what is resting membrane potential? Okay. When a neuron is at rest, meaning it's not conducting what we call action potentials, or we can say the neuron is not activated, it is at rest. Okay? And at rest, we have a certain setup here in the extracellular fluid, which is outside the cell, and the intracellular fluid, or ICF, inside the cell. And it's a certain distribution of ions. And really what we have here in the extracellular fluid, or ECF, we have more sodium outside the cell, we have more calcium ions outside the cell, and then we have more chloride ions outside the cell. In contrast, inside the cell in the ICF, we have more potassium ions and then more proteins, which tend to have a negative charge overall, so they're anionic. Okay? And again, it's not to say we don't have some calcium inside the cell, we just have more outside the cell. Okay? Now, when we start talking about this, it's important to understand that if we have the right ion channels, any one of these ions, sodium, calcium, chloride, and potassium, can move across the membrane down their concentration gradient. But these proteins are very large, and so they're confined to the inside of the cell. Proteins will never move across the membrane in this fashion. And what this causes at rest is for there to be a higher positive charge outside the cell and then a negative charge inside the cell. So at rest, the inside of the cell is negative, and that's very important to understand. In fact, when we measure the resting membrane potential, which is really just a voltage, in physics, a potential is a voltage. The resting membrane potential is negative 70 millivolts, which I have right here. Negative 70 millivolts. Millivolts are the units of voltage, and notice that this number is negative, which means that the inside of the cell is negative, and that by default means the outside is positive. Okay. Now, I'm going to go ahead and preface this that if we want an action potential to be conducted along the axon, okay, meaning we want this neuron to become activated and then do some action, then the action potential has to be propagated down this axon, but this resting membrane potential is going to have to move out of rest and go from negative 70 millivolts to about negative 55 millivolts. And those are two numbers that are really good to have memorized for neurons. Negative 70 is rest, and then in order to become activated, you have to get this up to negative 55 millivolts. Now, this is kind of some math stuff here, okay, but remember, negative 55 is actually more positive than negative 70. Okay, because these are negatives. Negative 70 is very negative. And if we want to get this to negative 55 millivolts, then we're going to have to do something where the inside of the cell becomes a little bit more positive, right? And that's the job of graded potentials, okay? So here's the bottom line. If ion influx occurs such that the inside of the cell becomes more positive, this is called an excitatory postsynaptic potential, or an EPSP as we'll often call. So when we say excitatory postsynaptic potential, emphasis on excitatory, this means that it's going to tend to make the inside of the cell more positive. And it's just going to be little bits. Little bits. Like if we have some positive ions that move inside the cell and make the inside of the cell more positive, it might take it from -70 to -69 millivolts or -68. It's not going to be massive fluctuations, but you have to have a lot of these EPSPs summed together in order to get to -55 millivolts. Okay? Also, in contrast, if ion influx occurs such that the inside of the cell becomes more negative, this is called an inhibitory postsynaptic potential, or an IPSP. And IPSPs, as you might guess, would actually bring the membrane potential closer to -70 and sometimes even further, maybe -71, -72, and so forth. And that takes the membrane potential further away from threshold, which is our -55. That's what we're trying to get to. And so the key here is, if we want to get to -55 millivolts, we have to do something inside here that makes the inside of the cell more positive. All right, so on this slide, we're now going to answer the question, how do we go from resting membrane potential of -70 millivolts and ultimately get toward -55 millivolts, which is going to be the threshold of excitation, in which case then we'll get an action potential along this axon. Okay? Now, let's think about this. With this picture right here, how could we basically make the inside of the cell more positive? Well, there's really only two options here. Okay? We could either have sodium influx, and by the way, the term influx means to move into the cell. That would certainly make the inside more positive. Our other option is for calcium to influx. Okay? And if calcium moves in, that would also make the inside of the cell more positive, because in both cases you're actually moving positive charges into the cell. And so what we're going to see here is that we're going to have some mechanisms in order to get calcium into the cell and then also sodium into the cell. And this is going to be the job of these proteins right here, specifically these blue ones, and also neurotransmitters, which I've abbreviated NT here. So let's take a look at this one up here, just for reference. So this blue protein right here, it's really representing two things. It's representing a receptor, that is a receptor for a neurotransmitter, and also an ion channel. Okay? So what we've got here is we've got this neurotransmitter that's going to be floating around, right? And it's going to bind to this receptor. Okay? And when it binds to this receptor, then perhaps a calcium ion can influx. So calcium will then move down its concentration gradient through the ion channel into the cell body right here, into the cell. And that will make the cell just a little bit positive, right? Because if we just move one calcium ion in, 1, 2, or even 5 or 10, that's just a little bit. That's not going to make it much more positive, okay? But every time I move one positive ion in here, that's going to be what we call a small graded depolarization. It's not a massive depolarization like we're going to see in the action potential, but we're going to get little bits of positive charge that are moving into the cell, and if we add those together or do what we call summation, sum them together, maybe we'll have enough positive charges at some point to get all the way up to -55 millivolts. Okay, we've got a similar case down here. We've got another one of these proteins, so maybe this neurotransmitter is floating around, right? Comes over here and binds to the receptor, then the ion channel that it controls open, and maybe a sodium moves in. Sodium's also a positively charged ion, and so this collectively will make the inside of the cell more positive. And so we start to get a bunch of positive charges that build up inside the cell, and so what we're doing is we're summing together all of these small graded depolarizations, or we're summing together these EPSPs, excitatory postsynaptic potentials. And really these are kind of the same thing. The small graded depolarizations, they are excitatory postsynaptic potentials. Okay? It's just a complicated way of saying we're just little bits at a time making the inside of the cell more positive. Now notice in this case I've only activated a couple of these ion channels, right? And so I didn't get to threshold yet. I just got up to -60. I would actually have to make it 5 more positive, in other words, to get to -55. So I'm not there yet. And if I don't get to that threshold, nothing happens down the axon. Okay? Even though I've made the inside of the cell more negative here, I have not hit the threshold, which is -55 millivolts. Again, a very good number to have memorized. Okay? Now let's suppose instead of just these two, now I've got a bunch more of these activated, okay? Now I've got even more positive ions moving in, whether it's calcium or sodium, right? And now I've got a lot more positive charges on the inside of the cell, okay? And maybe at this point, there's enough of these excitatory postsynaptic potentials, or EPSPs, that are now summed together that now I've got this membrane potential at negative 55 millivolts. And now that I'm at the threshold, I don't need to go any more than that. Just getting to the threshold will now trigger an action potential down the axon. Okay? So when we're talking about graded potentials, specifically the EPSP type, the excitatory ones, their whole job is to cause the cell body and the dendrites, of course, little bits at a time to get more and more positive. And when they get positive enough, that is to negative 55 millivolts, then you get an action potential going down this axon. Okay? So you either get to negative 55 or you don't. Okay? But you have to get to this threshold number. Okay? And again, these neurotransmitters would have to bind to these receptors for the ion channels. The ion channels would have to open, and then these positive charges would have to influx. That is calcium and sodium. Okay. Now, I just want to make this also very clear. Suppose that we've got this neurotransmitter, right? And it's not bound. Okay. What's going to happen if it's not bound is we don't get any ion influx. Okay. If there's no neurotransmitter bound to the receptor, then clearly there's going to be no ion channel that's open. Okay? So you do have to have that neurotransmitter there bound. That's very important. These ions aren't just going in spontaneously. Okay? But in this case, we got to threshold. So before we get into the inhibitory postsynaptic potentials and what those do, I want to just go over a few specific examples here of receptors and neurotransmitters, just so you know this isn't just a bunch of abstract concepts. These things are very well known. For example, this one right here, this is the glutamate NMDA receptor. Okay? This is a receptor that binds glutamate. So glutamate's floating around here, right? This is a neurotransmitter. And it'll come over here and bind to the receptor, and that will allow calcium influx. And so in this way, when glutamate binds to its NMDA receptor, that causes EPSPs, right? As long as that glutamate is bound. I got the same thing down here. Right here we have a different type of glutamate receptor. This one's called a kainate receptor. Okay? So glutamate again is floating around here as a neurotransmitter. It then binds here to the receptor, and then sodium ions influx, and that creates EPSPs, making the inside of the cell more positive. Okay? In other types of neurons, we might have an adrenergic receptor. These are receptors that bind catecholamines, such as norepinephrine, like this. Norepinephrine might be floating around here, or in some cases epinephrine. Norepinephrine binds, and that activates the adrenergic receptor, and then perhaps calcium ions influx. And so what you can see here is these are specific neurotransmitters binding to specific receptors that control specific ion channels. But in these cases, we have positive charges that are influxing into the cell body or the dendrites, and that's making them more positive. And overall, all those positive charges are summed together. And if they sum enough to get the voltage of the cell to negative 55 millivolts, then you get an action potential. Okay? Now, these are the jobs of these specific neurotransmitters, the ones that control EPSPs. But what about IPSPs? Okay? Now again, we talked about the distribution of ions here at rest. So we're back to rest again. Okay? So let's think about this. We've got sodium and calcium on the outside of the cell, chloride's also on the outside of the cell, and then potassium ions are on the inside of the cell. Well, let's think about what ways could we actually make the inside of the cell more negative. We looked at how do we make it more positive. How could we make it more negative? Well, here's the answer to our question. The most obvious one of these is to just influx chloride. Chloride is a negatively charged ion. It's an anion. And so if we move chloride into the cell, that would clearly make the inside of the cell more negative. Okay? Proteins are stuck here. Okay? They actually contribute to the negative charge that's there already, but they don't move, so we can't do anything with these. We also can't do anything with sodium and calcium. but we can do something with potassium. Potassium is a positively charged cation, but notice it's on the inside of the cell. So if we just move it out, which is actually called efflux, if we efflux potassium, then we're losing positive charge on the inside, and that will also make the inside of the cell more negative. Okay, so hopefully that makes sense. So let's look at these proteins here. And what we're going to see is that they pretty much are going to behave very similarly to the ones we just looked at, except they're going in the opposite direction. Okay, so let's look at this one right here. So this also represents a receptor and an ion channel. Okay, and we've got this neurotransmitter that's floating around. Okay, but this type of neurotransmitter, whatever it happens to be, is going to bind to this receptor that controls this ion channel. The ion channel is going to open, but then potassium is going to efflux. So this ion channel allows potassium to move down its concentration gradient, which is moving from inside to out. And since you'd be losing positive charges in this case, the inside of the cell is going to get just a little bit more negative. Okay? It's not a lot, okay? But you have to have little bits and a lot of them. Okay? And every time potassium effluxes, those little hyperpolarizations are called graded hyperpolarizations because we're making the inside of the cell more negative. And in fact, those small graded hyperpolarizations are now called IPSPs, or inhibitory postsynaptic potentials. Okay? Let's look at these over here. We've got this neurotransmitter floating around. Eventually it's going to bind to this receptor. This ion channel is going to open, and chloride this time is going to influx. And since we're moving a negatively charged anion into the cell, the inside of the cell becomes more negative. Okay? And every time a chloride ion influxes, or every time a potassium ion effluxes, the inside of the cell gets a little bit more negative. And so basically you're having a bunch of inhibitory postsynaptic potentials. Now overall, as long as you're keeping a neuron hyperpolarized, that keeps the neuron inhibited, and so it won't be activated. And so these mechanisms, in contrast to producing an action potential, these will actually prevent a neuron from having an action potential, and in some cases that's necessary. In some cases you do want that. You want there to be no activity of that neuron, and so you'll have these inhibitory neurotransmitters that keep the neuron inhibited, preventing the cell from having an action potential. And notice the membrane potential here. These inhibitory postsynaptic potentials bring it closer to rest, basically -70, and in some cases can make it even a little bit less, like -71, -72, and so on and so forth. Okay? And again, let's look at some specific examples, because this is again something that's very well characterized, and probably the most well characterized of all the inhibitory neurotransmitters is GABA. And GABA has two kinds of receptors, a GABA A receptor and a GABA B. So GABA A, which is shown right here, obviously is going to bind GABA, and so GABA, which is a neurotransmitter, is going to be floating around. Eventually it'll bind to the GABA A receptor, and again this controls an ion channel specifically for chloride, and so chloride will influx. Okay? And then that means you get more negative charges on the inside of the cell. Okay? Another kind we can have is GABA B. GABA B also binds GABA, so GABA is floating around here. GABA binds to the GABA B receptor, but in this case it controls a potassium channel, and so potassium effluxes. That's removing positive charge from the inside of the cell, which overall makes the inside of the cell more negative. Okay, GABA is known as the principal inhibitory central nervous system neurotransmitter. This is actually why you'll see GABA supplements in the store or the drugstore. People usually take them with the hope that they'll actually calm the brain, inhibit certain parts so that you can actually fall asleep at night. And there's some limited evidence that that actually can work. Okay? In contrast, glutamate, which we talked about for EPSPs, that activates the neuron, glutamate is the principal excitatory central nervous system neurotransmitter. And so generally in the brain, when you want to activate neurons, glutamate is used. When the brain wants to inhibit neurons, GABA is generally used. Okay? So hopefully this makes sense. And overall the key is, if you want a neuron inhibited, you use IPSPs. And to do that, you use inhibitory neurotransmitters that produce IPSPs, that make the inside of the cell more negative. But if you want the neuron to become activated and produce an action potential, you need EPSPs. okay, which collectively make the inside of the cell more positive, and if they sum enough to be negative 55 millivolts, you now get an action potential going along the axon, and that can lead to the activation of another neuron, and we're going to talk about all that physiology in future videos. But I just wanted to make sure that graded potentials make sense to you, because I think by looking at these little tables that compare them to action potentials, we get lost in what they actually are. The graded potentials are just a bunch of small depolarizations and hyperpolarizations that are always added together, and if they get positive enough, if you have enough of those depolarizations, which means enough of these EPSPs, you get to negative 55, you get an action potential. So we're going to pick up in the next video talking about the action potential that goes down the axon of these neurons. Please make sure to like this video and subscribe to my channel for future videos and notifications. Thank you very much.